All right, question number 52, local invasion and metastasis. The first thing we're going to talk about is what the difference between invasion and metastasis actually is. And there are obviously two very different terms which you need to be able to define properly in your exam. So, invasion is a term with respect to tumor that is used to describe direct extension or penetration by tumor cells into neighboring tissue. Whereas metastasis is simply the spread of tumors to sites that are physically discontinuous from the primary tumor. And that metastasis is technically a marker of malignancy for a tumor. So if a tumor does metastasize, it marks that the tumor is malignant. And so if you were to imagine that I have a tumor mass here and I have neighboring tissue here. If these cells are able to extend into this neighboring tissue while still being connected with the primary mass, with the primary tumor, a physical connection between them still exists, it will be termed as an invasion. But if I have a tumor that I've identified, for example, as lung carcinoma in the lungs, and I find that these cells are also present in the adrenal glands, physically discontinuous, they have absolutely no connection to one another, this is when we term the process as metastasis. And so that is an important distinction that you need to be able to make. The next thing we need to talk about are what are some of the features of a tumor that you will see, whether those are microscopic features or macroscopic features that will cue you into thinking that yes, this tumor does have metastatic potential. And so the first thing is at the microscopic level, you will see that the cells of the tumor lack any form of differentiation. The more differentiated a tumor cell is, meaning the more it resembles the parent tissue cell type that it is related to, the less likely it is to metastasize. If it has good differentiation, that means it has taken up, it not necessarily taken up the functionality, but it resembles the parent cell type, it is less likely to metastasize. However, if it is poorly differentiated, meaning it lacks differentiation, this can be a cue for us to think that this tumor can have metastatic and thus malignancy potential. Aggressive course of invasion into nearby tissues is another sign that a tumor has high metastatic potential. Other important features, relatively easy to think about actually, are just whether the tumor has rapid growth or whether it is large in size. Of course, a tumor can have these properties and still not metastasize at all, but if these features are present, you're more often than not more likely, there's a very high probability that the tumor will metastasize. And so now we're going to talk about the process of metastasis, which is also known as the metastatic cascade, and I will go through it in the form of a flowchart. And so at the basis of every tumor formation in general, we have obviously genetic and epigenetic alterations taking place in some sort of cells. And so an accumulation of complementary genetic plus epigenetic factors or alterations in some cells grants this tumor mass a metastatic phenotype. You don't really need to know into too much detail which genes are associated with metastasis or whatnot. I will mention a few mutations that are of relative importance for you to be able to mention, but it will be completely fine if you do not. So, what happens after that? What happens when the tumor has this metastatic phenotype? We can now expect the progression of the metastatic cascade. And so the first step in this process is the invasion of the extracellular matrix. And so we've got a few cells here. We've got the basement membrane of these cells. And once these cells are able to penetrate through the basement membrane and into the extracellular matrix, that is normally the first step that is associated with the metastatic cascade. This process is mediated by proteolytic enzymes. You don't really need to know what these proteolytic enzymes are, but for your knowledge, I will mention them. It includes cathepsin D, it includes plasminogen activator, among other enzymes. There's really not too much you need to know. And now we're going to discuss this process in a little bit more detail. Invasion into the extracellular matrix, the degradation of the bone, the basement membrane is something we're just going to discuss in a little bit more detail now. How does this take place? Firstly, you have mutations in E-cadherin transmembrane glycoproteins. And these glycoproteins, these E-cadherin transmembrane glycoproteins, are responsible for the homotypic adhesion between cells. And so if you have a mutation in the E-cadherin transmembrane glycoprotein, this basically means that you have a loss of E-cadherin structure, and that thus ends up meaning that you end up lacking cell-to-cell adhesion. And a little bit of a brief overview of what's happening once this process takes place, just for your understanding, is that we have a process occurring which is known as epithelial mesenchymal transition. And so this basically means from histology, you must remember the basic properties of epithelial cells and of the basic properties of mesenchyme. I am just gonna very simply define the way I understood it, because histology was not my greatest subject by any means. But as far as epithelium is concerned, it consists of cells that have high polarity and an organized structure as such, right? As compared to a mesenchymal type of cell, it can simply be basically defined as, in a way, connective tissue that consists of cells that lack polarity and strong adhesions, suspended in high amounts of extracellular matrix ground substance, right? And this is not really supposed to be a histology class for you, but what I really wanted to understand is that normally the cells of a tumor, right, in this case we're talking about epithelial tumors, that these cells undergo what we call an epithelial mesenchymal transition, meaning that they lose their polarity, they lose their adhesion because of this E-cadherin transmembrane glycoprotein mutation. And so I mentioned that I would just let you know some of the important metastatic genes. You don't really obviously have to be able to mention them in the exam, but just for you to know. For this epithelial mesenchymal transition to take place, you need to have down regulation of epithelial markers, and you need to have up regulation of mesenchymal markers. And these two processes are associated with mutations that occur in transcription factors SNAIL, SNAIL, whereas changes that occur resulting in increased regulation of mesenchymal properties and mesenchymal marker properties are associated with changes in transcription factor twist. Just mentioning these for you. Please do not worry too much about it. This was just to help you get an overall picture of what is actually happening at the cellular level, at the very basic level, at the very microscopic level, for you to understand what changes are taking place that allow for a cell to be able to lose its adherence, for the cell to completely dissociate from this tumor mass and metastasize. You don't really have to worry about the individual markers I've mentioned, but for your knowledge, just so you have a whole picture, a clear picture of everything that's going on, this was important to mention. Please do not worry too much about it, and if of course it does not make sense, just write to me. You all know who I am, I hope. Once all of this is done with, and a secondary step that occurs is now the activation of type 4 collagenases that break down basement membrane and allow for cells to migrate into the extracellular matrix. And the proteins that allow for this are basically what we call matrix metalloproteases. All right. So what's the third step that occurs? Now, one thing that is very important to understand with respect to this entire process is that a metastatic cancer normally has a lot of mutations taking place and a lot of loss of regular cellular functions that actually allow it to metastasize. And so the third thing we are going to discuss in this metastatic cascade is when there is a change in expression. I'll actually do this in red for consistency. Third step is a change in expression of antigens on tumor cells. And due to the changes in the expression of these antigens, the tumor cells, instead of specifically adhering to each other, can now adhere better to cells of the extracellular matrix. And all of this, basically, this entire process allows for the tumor, it aids in the process of locomotion. Now, the process of locomotion can further be aided by chemokines and growth factors that are secreted by the tumor cell. And these chemokines and growth factors cause autocrine signaling for the tumor. And obviously, if you're asked why the tumor can produce these chemokines and growth factors to begin with in the first place, because normal cells normally don't do this, it's simply because of the truckload of genetic and epigenetic changes that are taking place that grants the cells of a tumor these abilities, which would otherwise not normally be present in the cell. Now, following the cells of the tumor being granted the ability to, for locomotion to take place, what's going to happen next is the process of intravasation. which basically means that the tumor cells that are now that we have been talking about all this while can now enter the bloodstream. And so there are two ways this process can happen. The first way is that we can have a direct digestion of already present vascular channels, so digestion of vascular endothelial cells can introduce the tumor into the bloodstream. Or the second way that this process can take place is simply that the tumor at hand produces growth factors that result in angiogenesis. And so you'd essentially imagine that the tumor grows its own blood supply, which it can then invade too. And this blood supply obviously is, because how VEGF works is that you basically end up sprouting new vessels from already existing vessels, and so you already have a bridge and a connection for the tumor to get into the bloodstream. And obviously the process of intravasation is not limited to the blood, so this basically means the spread of these tumors now can be hematogenous, or it can be lymphatic. And so you already know that some tumors are more likely to metastasize through the lymphatic channels, and the others are more likely to metastasize through hematogenous channels. We'll discuss which tumors as a group are able to do it in which specific way. But for now, just remember intravasation as the step that we're involved in. And another important thing that is worth mentioning is that the tumor cells normally travel as, now once they're into the bloodstream, they migrate as multicellular aggregates. And multicellular aggregates can be homotypic in nature, which basically means that the migration, like the tumor mass that we're talking about that is now undergoing locomotion and migration, consists only of tumor cells. Or it can be heterotypic, which basically means the mass we're concerned with now includes tumor plus blood elements. That's something important to know. Now, once all of this is done, once the tumor is into the bloodstream, what happens next? We now have circulating tumor cells. What is going to happen now? The first important thing is to talk about where the tumor can end up going. And so the location and drainage, or the location and the vascular drainage of the tumor can be to its nearest organ, so to the nearest organ. It can, if it's spreading by a lymphatic channel, it can be to the nearest lymph node that drains the organ. And obviously the first lymph node in which metastatic cells are present in, or the ones they first metastasize to, is known as the sentinel lymph node. Of course, post that it can go into other lymph nodes as well. Now, there are obviously some mechanisms by which these circulating tumor cells need to be able to avoid the immune system, because ideally the immune system should be able to, catch or, surveillance through the blood and find any tumor cells or any irregular cells and be able to destroy them. And so for a tumor to be circulating in the blood, it has to have some mechanisms to escape from tumor dormancy and immunosurveillance. And this is usually mediated by an expression of CD4 CD47 cells, which is basically a signal to immune cells that, don't come after me. It's a don't eat me signal. That's the way I learned it, at least, and I remember reading this in Tomer's notes, and that's how I learned it. So, yeah, CD47 is an important surface antigen that these circulating tumor cells present with that helps them to evade the immune system. The next thing that we need to talk about is the tropism of tumor cells for specific tissue. So, tumor cell tropism. And this basically means that a tumor cell that has adhesion molecules whose ligands are found in specific cells of certain organs is very indicative of the fact, is rather very prognostic of the fact of where this tumor will end up metastasizing. So you imagine if I have an adhesion molecule for which ligands are found in the bone, for example, the tumor is likely to metastasize to the bone. And so for a tumor to actually get out of the bloodstream and into its new site, its new favorable site, it has to undergo extravasation. And this ideally simply follows the same steps as in white blood cell leukocytosis in the case of inflammation. The steps, if you will remember, were rolling, adhesion, and then extravasation. And this entire process normally ends up utilizing the same integrin and surface receptors that are present on endothelial cells for extravasation. Right, and once all of this is done, the last thing is the tumor ends up colonizing its metastatic site. It's the new site where this tumor is now arrested in. What happens for this colonization process to take place is that once the tumor is arrested in its favorable site, it will secrete growth factors and cytokines that will act on stromal cells, local stromal cells, and will end up making this environment hospitable or habitable for these tumor cells. Right? And that pretty much sums up what the metastatic cascade was. Just to summarize over here really quickly in this very poorly drawn square, the 5 steps that you need to be able to talk about are invasion. We defined what it is. Then intravasation, then circulation, followed by extravasation, and then finally colonizing the new organ that the tumor cells end up in. And so one last thing that we need to discuss now is obviously the roots of metastasis we already mentioned are lymphatic and hematogenous, or just simply, that a simple seeding of these cancer cells. But what we need to talk about is for which type of cancer cells is a specific pathway more relevant. And so what we need to know is that lymphatic metastasis is the most common pathway for epithelial tumors. And the hematogenous pathway is more common for mesenchymal tumors. And normally hematogenous metastasis occurs through venous drainage as compared to arterial drainage, simply because the walls of the venous vessels are relatively thinner and they're easier to digest into and for metastasis to occur. And the other important thing is there are some exceptions to this, and that basically means that there are some carcinomas that are able to metastasize hematogenously, and you should know them. It's as simple as that. The four that are pertinent to you knowing are follicular thyroid carcinomas, choriocarcinomas, renal cell carcinomas, and hepatocellular carcinomas. And there are two theories that are used to describe why a cancer cell metastasizes to the location it does. And we already discussed this a little bit, but just to summarize again and quote again, the two major reasons why a cancer cell actually metastasizes to its actual location. First, it's simply because the new organ it goes to is the first capillary bed that is encountered by the tumor cells that are metastasizing. And the second is simply because some environments are more adapted or better suited for the survival of the tumor cells, and this, like I said, had to do with the fact that there are certain adhesion molecules that are present on the surface of these cells for which ligands are found in specific sites. So adhesion plus ligand interaction only occurs at specific sites. And so the last thing we need to discuss really quickly are some common sites of metastasis. First common site is obviously regional lymph nodes. Second is the liver. Third, metastasis to bones, and so most of the about 50 to more than 50 rather, a high percentage of bone tumors that are found are not primary bone tumors. They are metastatic tumors, and these include tumors that arise from the prostate, the breast, the lungs, and the thyroid. And then, of course, about 50 to 60% of the tumors that are in the brain are also metastatic, and these arise from the skin, the colon, the breast, and the lungs. So these are some of the common sites of where a tumor cell will metastasize to, and that should sum up this entire question.